Okay, imagine for a second a future where we don't just, launch pre-built metal cans to other planets piece by expensive piece. What if we could actually grow our homes out there? Like, literally, from the ground up, using the alien soil itself.
It sounds completely like science fiction, doesn't it? But that's exactly the wild idea we're diving into today. It's called Myco-Architectural Generative In Situ Construction, or MAGI construction for short.
Magi construction. I like it. And their motto is pretty bold, too.
Oh yeah, it sums it all up. We don't build on new worlds, we grow them.
Okay, so our mission for you today is to really unpack this pretty unique scientific proposal. It comes from Shahryar Ghiasi. He's an Iranian architect, city management expert, and, well, clearly a big science thinker.
Right. We're going to pull out the key insights from his work. I want to show you how these genetically engineered fungi could totally flip the script on living off-world.
Offering shelters that aren't just static boxes, but living things. Self-healing, protective.
Exactly. Truly protective environments for astronauts. It's a whole new way of thinking about space settlement, really. A genuine paradigm shift.
So let's get into that vision. Growing our future in space. You said it's not about setting up huge pre-made habitats. So what is the core concept here? What does Ghiasi actually propose sending?
Well, instead of tons and tons of metal, he suggests launching these surprisingly light seed packages. Think really compact.
Seed packages. Containing what?
Inside, you'd have dormant fungal spores, seeds, basically, and a swarm of small, compact robots.
Okay.
And here's the kicker. The vast majority of the final habitat's mass, maybe over 90%, isn't launched at all. It's sourced right there at the destination.
From Martian soil, you mean, or asteroid dust?
Exactly. Martian regolith, asteroid material, whatever's available locally. That's the raw building block.
Wow. So yeah, that's not just creaking the old way. It's a totally different approach. Moving away from that fabricate and ship model we've used forever.
Completely. Ghiasi calls it the seed and cultivate model. It reframes how we even see other planets.
How so?
Instead of just hostile, empty places we need to protect ourselves from, we start seeing them as resource nurseries. Places full of raw materials just waiting to be cultivated.
Resource nurseries. I like that. It implies potential, not just danger. And obviously that massively cuts down on launch costs and complexity, right?
Oh, hugely. The cost difference could be enormous.
Yeah.
An order of magnitude, potentially.
Okay, this sounds amazing. Almost too good to be true. How does this growing actually happen? Ghiasi talks about 3 pillars, right? Let's start with the first. Mycology, the living scaffolding.
Right. Mycology, the study of fungi. The star here is mycelium.
That's the root network, yeah.
Exactly, the thread-like root structure of fungi. And its properties are just perfect for this. It grows incredibly fast, it's surprisingly strong, and very lightweight.
And you can shape it.
You can. It forms this dense interwoven mat, like a natural fiber network, that can be guided to grow into specific forms.
What's it made of?
Oh,
What
yeah.
gives it that strength?
It's mainly chitin, and the same tough stuff you find in insect exoskeletons or crustacean shells.
Okay, so it's naturally strong.
Very. And when it's combined with other materials, like regolith, its tensile strength is impressive. Some tests show it beating reinforced concrete in compression.
Wow. Okay, so it's not just the threads tangling up, right? There's something more going on to make this mycostone so strong.
Absolutely. There's a process called bioconcretion.
Yeah.
The fungal hyphae, those individual threads, they don't just trap the regolith particles. They actually secrete enzymes and glycoproteins.
Like a biological glue.
Sort of, but even more integrated.
Yeah.
These secretions partially dissolve the surface of the regolith particles and then bind with them chemically at a molecular level. It fuses everything together into this solid, continuous mass, a real mycostone.
Fused together organically. And Ghiasi mentions it's not uniform, it's a functional gradient material. What does that mean?
Yeah, that's a really clever part of the design. By controlling the growth conditions like nutrients, humidity, gases, you can make the structure develop different properties at different depths.
Like layers in an onion.
Exactly like that. So the outermost layer, the crust, could be deliberately hardened, maybe using robotic solar concentrators to basically bake or vitrify it into a tough, glassy, non-porous shell. Maximum protection from the outside.
Okay, a hard outer shell. Then what's inside?
Beneath that crust, you have the shield layer. This is thicker, it's still living mycelium, and it's engineered to be rich in melanin.
Melanin for radiation protection.
Precisely. Melanin is great at absorbing radiation. This layer would also hold water, adding even more shielding, and it's where the main self-repair happens. It's the living, functional core.
And the innermost layer, the part astronauts interact with.
That's the interface. This layer is engineered for human comfort and function. It could be softer, maybe integrated with things like bioluminescent algae for gentle, natural lighting.
Bioluminescence.
Mm.
Wow.
Yeah, or biofilters for air quality. The texture could be made more pleasant, less like rock, more organic. The goal is a psychologically supportive environment, not just a functional box.
That makes so much sense. Okay, amazing. That leads us to the second pillar, synthetic biology, the engineered blueprint. So this isn't just garden variety mushrooms we're talking about.
Oh, definitely not. Think of the fungal spores as the highly advanced software driving this whole process. They'd start with robust, fast-growing fungal species, sure, but then they'd be heavily modified using synthetic biology.
Genetically engineered for space.
Exactly. Engineered to thrive in alien conditions. Key traits would include the ability to actively incorporate local minerals, like Martian regolith, directly into their cell walls.
Making them stronger and better at radiation absorption.
Right, and maybe they could even be programmed to produce useful things on site, biofuels maybe, or light, like we mentioned, even medically useful molecules for the crew.
That's incredible. But what about other capabilities? You mentioned radiation.
Yeah, this is where it gets really wild. Ghiasi talks about radiotrophic adaptation, meaning potentially borrowing genes from fungi, found near Chernobyl fungi, that actually use melanin to convert gamma radiation into chemical energy.
Wait, they eat radiation?
Essentially, yes.
Yeah.
They harness it. So the
Yeah.
habitat wouldn't just block radiation, it could potentially turn that thread into a source of energy for the fungus. It's like biological alchemy.
Turning danger into fuel. That's mind-bending. So it's building, shielding, and maybe even producing power.
And resources. You could add genes for enzymes that break down specific Martian minerals, like iron oxides for extra strength, or even those toxic perchlorates everyone worries about on Mars.
Neutralizing toxins while building?
Potentially, yes.
Wow.
Neutralizing them or maybe even using them as an energy source. And for life support, the fungus isn't alone. It acts as a scaffold.
A scaffold for what?
For a whole micro-ecosystem. You could co-culture engineered cyanobacteria within the inner walls. They'd consume the Curio astronauts breathe out.
And produce oxygen.
Oxygen, and maybe even some basic carbohydrates or nutrients, creating a closed-loop system right there in the walls.
That is true symbiosis. It sounds fantastic, but okay, the big question, safety. What stops these superfungi from, escaping and growing everywhere? Planetary protection is a huge deal.
Absolutely critical question. And the proposal has a built-in answer. Programmed apoptosis.
A kill switch.
Basically, yes. Multiple kill switches, really. The fungus would be engineered with genetic circuits that trigger cell death under specific conditions.
Like what?
Like exposure to, say, a certain frequency of blue light that doesn't exist naturally in its intended Martian cave or habitat environment.
Right.
Or if it loses the specific nutrient feed provided by the robot.
So if it gets out or if the system fails, it just dies off.
That's the idea. It's designed to be contained. A biological failsafe to prevent uncontrolled spread and ensure we're responsible explorers.
That's reassuring. Okay, that brings us logically to pillar 3, the robotic swarms, the interstellar gardeners, as you call them. So not construction bots in the usual sense.
Not at all. Their role isn't hammering and welding. It's much more subtle. They are the gardeners, the tenders, the systems integrators. They manage the biological growth.
Okay, so paint a picture. What are they actually doing day to day on Mars?
Well, first up is deployment. They land, they set the initial template, maybe an inflatable bladder, maybe a simple frame defining the habitat's a basic shape.
Got it. The mold, essentially.
Pretty much. Then they start gathering local regolith and water ice, mixing it into a kind of nutrient-rich paste or slurry, and applying it over that template.
Okay, preparing the soil bed.
Exactly. And then they inoculate it with the fungal spores. As the mycelium starts growing, the robots shift into nurture mode. They use sensors, multispectral sensors, to monitor everything. Growth rate, density, health.
And they feed it.
Precisely. They deliver water, nutrients, maybe localized warmth, exactly where it's needed. They can even guide the growth using stimuli like light fields or weak electrical fields, almost like pruning a bonsai tree, but on a massive scale.
Dieting the structure into shape. And what about integrating the hardware? Doors, windows, ports.
That's the crucial systems integration part. As the mycelial walls grow and start to solidify, the swarm carefully embeds the non-biological bits, airlock frames, window fixtures, data ports, power conduits.
And the mycelium just grows around them.
It's guided to grow tightly around them, forming perfect, seamless, airtight seals. And because it's living, those seals are potentially self-repairing too. It integrates the tech right into the living fabric of the building.
Imagine growing your airlock seal. Okay, so let's connect this back to the big challenges of space exploration: cost, sustainability. This MGI approach sounds like it could be a logistical game changer.
Oh, absolutely. Let's talk numbers, roughly. A traditional Mars habitat module might weigh, what, 8, 10 metric tons, maybe
That's
more.
heavy stuff to launch.
But a MGI seed kit, the spores, the small robot swarm, the initial bioreactor, the template that might only weigh 1 or 2 tons.
That's a massive difference.
It's a radical mass reduction.
Yeah.
And since launch cost is mostly about mass, you're potentially cutting the cost of establishing that first habitat by like 90%. Because, again, most of the final mass is sourced in situ.
From Mars itself. And it's not just cheaper for the first one, is it?
No, that's the other revolutionary part. Traditional expansion is linear. One launch, one module, build piece by piece.
Right. Slow and inexpensive.
But MGIs is biological. Growth can be exponential. A single thriving mycelial habitat isn't just a home. It's potentially a factory that can bud off or seed numerous new structures, connect tunnels, add labs, garages.
So the base can grow itself, essentially.
Exactly. It's a self-propagating system potentially, which also means fewer complex launches needed overall.
Okay, economics aside, what about keeping humans safe and healthy? Space is brutal. Radiation, micrometeoroids.
Big advantages there, too. For radiation, that thick 1-meter-plus wall of melanin-rich, water-infused mycelium and regolith composite that offers potentially far better shielding than the thin metal walls of current spacecraft or habitats.
Better than aluminum.
Potentially much better, especially against galactic cosmic rays. And then there's the self-healing. This is huge.
Right, you mentioned that. So a small impact.
A micrometeoroid pings the habitat. Today, that might mean a dangerous, complex spacewalk and EVA to patch it. With MAGI, sensors detect the breach, the robotic swarm targets the area with nutrients.
And the mycelium just regrows, seals the hole.
Within days, potentially. The living wall heals itself. That drastically improves safety and reduces astronaut workload. It's a game changer.
It really is. And what about the inside, the psychological side? Living in a metal box for years sounds rough.
It's a known challenge. The isolation, the sterile environment, it takes a toll.
Yeah.
Compare that to a MAGI habitat.
What would that feel like?
Imagine living inside something with gentle curves, maybe soft ambient light from bioluminescent patches in the wall, perhaps a faint earthy smell, stable humidity. It taps into that concept of biophilia.
Our innate connection to nature.
Exactly. That connection to a living, natural-seeming environment could significantly boost mental health, reduce stress, and just make long-duration missions more sustainable for the crew. It's about thriving, not just surviving.
Living with the environment, not just in it,
Yeah.
which links perfectly to ISRU in situ resource utilization. MGI seems like the ultimate expression of that idea.
It really is the epitome of ISRU. It's not just using local resources, it's fundamentally assimilating them.
How so?
Well, Martian Coraux from the atmosphere feeds the cyanobacteria in the walls. The regolith is the main structure. Local water ice provides the water needed for growth. It's taking the raw ingredients of Mars and weaving them into a living, functional system.
Truly living off the land. And thinking long term, what about space junk? Orbital debris is a growing problem.
Yeah, it's a mess up there. Now, MGI won't clean up existing debris, obviously, but it offers a more sustainable path forward. These habitats, especially if used for orbital stations or on asteroids, could be designed with that kill switch we talked about. So at the end of their useful life, they could be triggered to fully biodegrade.
Break down into harmless organic components.
Exactly. Instead of leaving behind chunks of metal posing collision risks for generations, you have something that returns to its basic organic constituents. It's just a more responsible way to build in space.
Okay, let's make this concrete. Ghiasi outlines a hypothetical Mars mission, Project Genesis. Can you walk us through how that might unfold?
Sure, it paints a really vivid picture. Step 1 is launch and arrival. A single rocket, relatively light, carries the Genesis seed, payload, the spores, robots, bladder, initial nutrients. It lands somewhere promising on Mars, maybe near known subsurface ice, like Arcadia Planitia.
Landed safely, then the robots wake up.
Pretty much. Month 1 is deployment. The lander deploys the robotic swarm. Their first job is to inflate maybe a 10-meter diameter spherical bladder, the template. At the same time, other robots start mining nearby regolith and ice.
Getting the materials ready, how long until we see growth?
The next phase, months 2 through 12, is inoculation and growth. The swarm mixes the regolith and ice water into that paste, coats the bladder, and injects the engineered spores. A small bioreactor gives them their initial kickstart.
Under a tent, you said.
Yeah, probably covered by a lightweight translucent tent to maintain the right temperature and humidity early on. Then the mycelium just takes off. colonizes the regolith matrix, growing several centimeters a week.
That's pretty fast for building a house. What happens next? Months 13 to 18. Integration and hardening.
Right. Now the walls are getting thick, maybe half a meter. The swarm starts carefully cutting openings in the now buried bladder and installing the hardware. Airlock frames, a science port, windows.
And a mycelium of seals around them.
It's guided to grow right up to them, creating those perfect hermetic seals. Simultaneously, maybe a specialized robot with a solar concentrator starts vitrifying the outermost couple of centimeters, baking it into that hard, protective crust.
Okay, nearly there. Months 19 to 24. Maturation and pressurization.
Yep. During this time, the cyanobacteria inside the inner wall layer really get going. They start processing the internal atmosphere, maybe initially filled with Martian co-ro pumped from outside, slowly converting it into a breathable oxygen-nitrogen mix.
So the habitat makes its own air.
That's the goal. By month 24, the structure is mature. It's maybe 1.5 meters thick in total, multi-layered, self-healing, radiation shielding. The internal bladder can be deflated and packed away for reuse. It's ready for inhabitants.
Wow. So month 25, the crew finally arrives. What do they find?
They dock their ship, open the hatch, and step into a fully formed, pressurized, breathable habitat. It's already humming along as part of the local Martian environment. Their first job isn't frantic construction. It's science and exploration, because their home was literally grown for them.
That image, arriving to a living base, it's fundamentally different, and Ghiasi imagines this expanding beyond just one dome, right?
Oh yeah, this is just the start. Phase 1. Initial settlement isn't just about the main habitat. You use AMAGI to grow connecting tunnels between modules, creating a larger, contiguous pressurized zone.
Like building a little village.
Exactly. And you could grow infrastructure too.
Mm.
Solid dust-free landing pads for rockets. That's a big deal on Mars. Maybe even paved pathways for rovers between structures.
Okay, so connecting everything up. What about phase 2? Base, expansion, and specialization.
Now you start growing purpose-built structures. Greenhouses, maybe with sections of wall engineered to be more translucent for optimal plant growth.
Growing food inside grown walls.
Right. Or unpressurized but shielded garages and workshops for equipment maintenance. Even dedicated waste processing units that are essentially living bioreactors, breaking down waste biologically.
And you mentioned lava tubes.
Yes, it seems ideal for that. If you find a stable lava tube, which offers fantastic natural radiation shielding, you could use MGI to line the porous rock walls, sealing them and creating large, easily pressurized volumes inside, like inhabiting a natural cave, but making it airtight and comfortable.
That sounds incredibly efficient. Okay, phase 3 gets even more ambitious. Advanced and orbital applications.
This is where the lines blur between habitat and factory. The walls themselves could become manufacturing platforms. Imagine engineering the mycelium to secrete useful substances directly from atmospheric carbon and local minerals.
Like what?
Pharmaceuticals the crew needs, specific nutritional proteins, maybe edible mushrooms growing right out of the wall, or biopolymers for 3D printing other items. The base itself becomes productive.
A biofactory. And beyond Mars.
Absolutely. Think about resource-rich asteroids. You could potentially grow habitats, fuel depots, maybe even structural components for spacecraft right there in microgravity using asteroid materials.
Growing infrastructure in the belt. And the biggest dream of all.
Terraforming. On a much, much longer time scale, of course. But you can imagine vast networks of specifically engineered fungi deployed across Mars, processing the toxic perchlorates in the soil, fixing atmospheric nitrogen and carbon, slowly creating the first layers of organic, fertile soil. Basically preparing the ground biologically for more complex plants later on. It's the first foundational step towards making Mars more Earth-like.
From building a home to building a biosphere.
Yeah.
Incredible. Okay, let's pull back to the big picture. Why does all this MGI stuff really matter? What are the core takeaways Ghiasi wants us to understand?
Well, boiling it down, it hits all the major pain points economically. It's about that radical mass reduction, slashing launch costs, and enabling potentially exponential self-replicating growth. That's huge.
Functionally.
Functionally, you get structures that are self-healing, adaptable, multi-purpose. They're the structure, the radiation shield, the insulation, potentially part of the life support, even a manufacturing base.
All in one.
And for the humans inside, the astronauts.
From a human-centric view, it offers better protection radiation, micrometeoroids, plus that crucial psychological benefit from biophilia, living in a more natural feeling space, and inherent fire retardancy, which is always vital.
Right. And ethically, sustainably.
Ethically, it's true. ISRU living with the land, not just on it. And that end-of-life biodegradability, it points towards a more responsible, sustainable way of expanding beyond Earth, leaving less junk behind.
It's a really holistic vision. Okay, a final thought for you, our listener, to chew on. If we can figure out how to literally grow our homes from the soil of another world, what other impossible problems could biology help us solve?
Yeah, think about that shift. From building things
Yeah.
to growing things. What could that mean not just for planets light years away, but maybe for how we build and live more sustainably right here on Earth?
Something to think about.